So today we're going to be talking about cells. So cells, when you think about cells, the first thing you might think of is that they are super tiny, right? You have to use a microscope in order to view almost all of them, not every cell, but for the most part, cells are so tiny that you just can't see them with the naked eye. And to really drive home just how small these things are, I have a simulation to show you. Unfortunately, Flash Player is no longer supported, so I can't show you this in real time, but this is a recording of me doing this before. What we have here are some things that we are used to seeing, right? So this here is the text on a piece of paper. This here is a coffee bean, right? And so what I'm going to do any second now is I'm going to zoom in, and what's happening here is that we are getting smaller and smaller. So essentially imaginary microscope zooming in. And so the very first cells, right, the largest cells are even smaller than a grain of salt, right? Those aren't even human cells. The largest of the human cells is a human egg. And the reason it's so big is because it's actually 4 cells joined into one. Most of our cells are much, much smaller even than that. Okay. So typical cell, typical skin cell, if we zoom in even farther, our red blood cells are anucleate, so they are super, super tiny. Chromosomes, these here are organelles, which happen to be approximately the size of bacterial cells, and there's an evolutionary reason for that we're not getting into, but these are the organelles we're going to be talking about today. Note that viruses are much, much smaller even than the tiny little organs within our own cells. Right, so very, very tiny, which is why we need particularly precise technology to fight these viruses. Okay. One of the organelles we're talking about a lot is a ribosome, right? So tRNA, phospholipids, glucose, all of these molecules you learned about yesterday. And finally, this here is an atom. So I just wanted to drive this point home of how small the things are that we are actually talking about today and over the next few days in this class. Okay, so again, much, much smaller than anything that you guys can see with the naked eye. Okay, and so why exactly is that the case? Well, cells are small, not purposely to be inconvenient, right? They're not on purpose microscopic. Cells are small because they need to have a large surface area to volume ratio. That is, they need to have a lot of space on the outside of their cell relative to the inside of the cell so that they can take in all of the things they need to survive, right? So nutrients, other types of components, ions into the cell, and they need to have a lot of entry points on the outside. And of course, they need to be able to get rid of a lot of waste pretty efficiently, right? So the more doors you have to enter the store, the more efficiently you can get into that store, right? And so just to add some numbers here, not that you're responsible for knowing these numbers, just proof of concept here. All of these images up here have the same volume, right? So 1 cube versus 8 cubes versus 64 cubes, all of them have the same volume, 64 square centimeters. Okay. However, when we make smaller and smaller and smaller structures, right, in this case cubes, what we're doing is we are increasing the total surface area. So how much space is on the outside of the cubes? Again, same amount of space on the inside to store whatever we're storing, or, in the case of cells, to metabolize things, to generate action potentials, etc. But when we make smaller and smaller cells, we are greatly increasing the surface area to volume ratio. So 1 cube has a 1.5 to 1 ratio. Whereas if we divide that same space into smaller and smaller units, the surface area to volume increases 4 times. Okay, so cells are tiny, right? We have taken this route to increase the surface area to volume ratio. Now there are, as I mentioned before, some cells that are larger that we can see with the naked eye. Human eggs are large just because they actually contain 4 cells all merged into one. Other eggs, right? So a chicken egg or whatever. Of course, you can see this with the naked eye, but the reason a chicken egg can be so big, but the rest of our cells must be so very tiny, is that that cell is loaded up with all of the materials it needs, right? All of the nutrients, all of the inputs it needs for its entire, 3-week, 4-week incubation period. Okay, so it doesn't need to have any nutrients entering. Also, when the chick hatches, that is when all the waste is going to exit. So there's nothing coming into the egg, nothing going out of the egg. So it doesn't need to be confined to this large surface area to volume ratio rule. Okay, so take home message here, cells are super tiny because we need to have a lot of plasma membrane, a lot of these phospholipids for the exchange of materials with the outside world. Okay, all of our cells are called eukaryotic cells. Okay, so our cells are therefore similar in a lot of ways to the cells of plants, fungi, protists. Right? So most of the things you think about as being alive are also eukaryotic cells, with the exception of prokaryotic cells like bacteria and archaea. As you can see, the average eukaryotic cell, even though it's super tiny, is much, much larger than a prokaryotic cell like this bacteria here, okay? Which hopefully you got to appreciate from my little video clip a moment ago. Okay, and so why are our cells bigger than that of a bacteria? Well, our cells are, as you might imagine, a lot more complex, right? And the way that we increase this complexity and therefore allow cells to specialize in sending electrical signals versus contracting versus filtering something is that we have different compartments within our cells. Right, so each of these compartments can specialize in a particular task and can therefore be more efficient. These compartments are called organelles or little organs. They are membrane-bound spaces. These membranes are composed of phospholipids as well. Right, so this here is one organelle, which is actually the nucleus. It is surrounded by phospholipids, the same as the outside of our cells are surrounded by phospholipids, right? So essentially this produces this selectively permeable barrier, which we're talking a lot about in a couple days. But this selectively permeable barrier allows some stuff to come in, some stuff to go out, but not everything, right? So we don't have everything in the world coming into these little compartments. It's only the stuff that we need. And again, this increases efficiency. Okay. As always, these organelles and these cells and these tissues, et cetera, work together. Right, so none of these things are working independently of one another. And hopefully when we walk through protein synthesis later in this lesson, you will see just how much these structures have to work together towards the common goal of making more cells, of completing the ultimate tasks of the cells. Okay, so at some point in your educational career, you have surely taken a biology class, at which point you probably saw something that looks like this, right? Hopefully no one, has PTSD over this image, but this is a eukaryotic cell. Today, we are going to walk through a lot of the different structures within this cell, the structures being organelles. Now, on a lecture exam, I am not going to give you this image and say, okay, what is this structure right here? That's not what the focus of this class is. Instead, for the lecture, you should focus on what does this do, right? How is it involved in protein synthesis? How is it involved in mitosis, in cell replication? Okay, so that is where we're going. For some context, I am going to include this image up in the corner of a lot of my slides, just so that you can see where these organelles are relative to one another. Okay, example 1. Here is that same cell. The first structure that we're going to talk about isn't an organelle at all, but instead it is surrounding the entire cell and surrounding the organelles themselves. This is the plasma membrane, right? So this arrow is indicating where exactly on the cell this is located. As you learned about in our biochemistry lecture, the plasma membrane is made up of phospholipid bilayers. Okay, so all of these little blue spheres and these white spheres here are representing the phosphate head. Remember that this is the hydrophilic component of the phospholipids. Okay, so the outside of this membrane and the inside of this membrane are perfectly happy, not to anthropomorphize, but they are comfortable being or interacting with water, right? Because we as humans, we as organisms on Earth are made up of mostly water. It makes sense to have membranes that can freely interact with watery components. Okay? Inside, right, kind of sandwiched in between, as I was saying earlier, are the lipids, right? So these fatty acids here. Okay? And so essentially anything that crosses over this barrier, right, has to be physically allowed into or out of the cell, because anything hydrophilic, anything that loves water, cannot get past these lipid tails, and anything hydrophobic, so anything fatty, right, cannot pass over these hydrophilic phosphate heads. Okay, so in this way, the plasma membrane, right, around the outside of the cell, and of course the phospholipids that are surrounding the organelles, regulate the entrance and exit of molecules. Right, we'll talk about this here soon, on Monday, I believe. But essentially, this membrane is therefore selectively permeable. Right, in order to allow substances in and out, we insert proteins, right, that are essentially channels for allowing the passage into and out of the cell. Okay. Now, the insertion of all of these proteins and other things, like this type of protein, these branched little green things here are carbohydrates, right? Each one of the little green spheres is actually representing a glucose or other type of monosaccharide. Okay, so these are actually producing kind of like our cell's fingerprint, right? Allowing us to see, is that our cell or is that something foreign? Also, what type of cell are you? Okay, so all of these different things that are kind of squished into our plasma membrane make our membrane look like a mosaic, right? So sometimes the plasma membrane is called a fluid mosaic. Mosaic because of all of these funny things that are embedded within it. Fluid because the phospholipids are just that, they are lipids. And so you can think about the surface of our cells kind of like vegetable oil, right? So vegetable oil kind of slips and slides around. It has that slippery consistency, and we want that. We want our cell membrane to be this vivacious, this moving entity, so that we can take this protein here and we can move it across the cell if we need to. Okay, so we absolutely need that. And of course, if you're eating lots of trans fats, our ability to have this fluid component of our fluid mosaic is inhibited to an extent. Okay. So again, plasma membrane made of phospholipids. It is selectively permeable. Hydrophilic and hydrophobic components prevent certain things from going in and out, and we have to specifically select substances for passage. Okay. Now, looking inside the cell, again, what we see is the phospholipid bilayer, right? The plasma membrane around the outside of our cell, and a series of other organelles within it. Now, we are going to walk through quite a few of these organelles, not just as a list, here's the name of the organelle, here's what it looks like, here's what it does, but instead I'm going to present this content to you kind of as a story, right? This is going to happen a lot of times throughout the semester, but essentially we are going to walk through a particular process so that you know what is the structure, what is its function, what is its larger role in the activity of the cell, the tissue, the organism. And so the process that we're going to walk through today is commonly called the central dogma to biology. That is, this is a cornerstone of understanding for your progress through A&P and through the rest of your careers. So this central dogma is essentially the process of protein synthesis. It starts in the nucleus, right? And I'm going to write this all out for you here in just a moment, but it starts here in the nucleus, we have DNA that is always stored in the nucleus. It is never allowed to come out of the nucleus. So in order to make a protein using the instructions stored within our DNA, we first have to make a copy of that DNA. Okay, so you can think about DNA as being an entire cookbook. Okay, the recipes in this cookbook allow us to make any of the proteins in our body, right? So anything that we are ever going to need is, the recipes are stored in the DNA. Now, of course, we don't want to lug around the big cookbook all the time. We only want a single recipe to make, cookies, to make brownies, whatever. So we actually make a copy, right? Write it out in the same language, and that copy is called mRNA. Now we have to process this mRNA a little bit, and finally the mRNA is allowed to leave the nucleus to be translated within the cytoplasm, within the rest of the cell. Okay, so using this copy of the recipe, not the original recipe book, but just the copy, we can read that copy, that little recipe, and with it, we can make a protein, right? So we're making brownies or we're making cookies, like some kind of end product is what this enzyme called the ribosome is responsible for doing, okay? So again, we are going to walk through this process over the next few slides. Maybe one moment here. Okay. Okay, so what I have for you here is kind of the end product of what we're going to be drawing out. So just like I did before, I'm going to draw here and we're going to follow the PowerPoint notes and draw out simultaneously, right? So I'm going to be kind of flipping back and forth. This is what we're shooting for. Note that this process, I have drawn out on 2 pieces of paper. Okay, so if you want to draw really big or leave a lot of space for notes, maybe 2 pieces of paper side by side is what you should shoot for. But it is more than possible to condense all of this information onto a single page. Okay, so that is what we are working towards. Okay, so the very first structure that we are going to be talking about here is the nucleus. So the nucleus is kind of the cell's brain, or where the cookbook essentially is stored. Okay, in our image here, we can see this big spherical purple structure here. That is the nucleus. Now, because it is this kind of the brain of the entire cell, this is where all of the content is stored for the entire existence of the cell's life. We protect the DNA with not 1 phospholipid bilayer, but 2 phospholipid bilayers. So here we can see 1 pair of phospholipids and another pair. And so collectively, this is called the nuclear envelope, right? So really, really important to protect the DNA at all costs. Okay. Now, of course, some things need to come into and out of the nucleus. And so we insert into this double phospholipid bilayer some fairly complex protein channels called nuclear pores. And these nuclear pores only allow certain types of molecules, so things like hormones, things like transcription factors that will like turn on a gene or essentially allow it to be copied so that we can make a protein from it. So these here are the nuclear pores. They will not allow DNA to leave the nucleus. Again, we need to keep that cookbook safe. Okay. Now DNA is actually stored in a form called chromatin. Right. So if we look at this image here, we can see that we don't have these, I don't know, you usually see chromosomes like this, right? So that is one particular chromosome, but really these molecules aren't stored in this like nice wound up form. Instead, they are stored in a form that's almost like cotton candy, just kind of like all over the place within the nucleus. And so that's a good thing because we can, or our enzymes can access individual genes on our DNA double helix at will, right? It's not embedded within this wound up chromosome form. Again, DNA cannot leave. It is way too big of a molecule, cannot pass through the nuclear pore. Okay, and so how exactly do we get the cookbook information from the nucleus out into the rest of the cell where we actually need it, where we can actually use it? We synthesize a copy of that DNA. So a particular gene, only one recipe, not the entire book, just one recipe, and that copy is called RNA. Right? The process that we're talking about is called transcription. So let me draw out for you here what we are talking about. Okay. All right, first, I am drawing part of the nucleus. Okay, remember that the nucleus has a double phospholipid bilayer. All right, so this here is called the nucleus. Okay, phospholipid bilayer, phospholipid bilayer. Okay, within that phospholipid bilayer, we have embedded specialized proteins called nuclear pores. Okay. These nuclear pores do not allow DNA out of the cell. They only let things like RNA and proteins and other important like transcription factors in and out of the nucleus. Okay. Now, keep in mind this is only part of the nucleus. All the way the other side of my page here is the edge of the cell. Okay, so this is the plasma membrane. Okay, remember that essentially this blue line is a phospholipid bilayer. Right, so here are some phosphate heads, 2 layers with lipid tails on the inside. Okay. So outside is hydrophilic. The inside is hydrophilic, and sandwiched in between is hydrophobic. So we'll come back to this over here. For now, entering into the cell all the way over here, the nucleus. Housed within the nucleus, as we know, is the DNA. The DNA is spread out all over the cell, or sorry, all over the nucleus, in its chromatin form. Okay. And so when I draw this green line, all right, so let's zoom in on this. What I am drawing, or what I'm implying, is the double helix of DNA. And essentially, this double helix is made up of nucleotides, A's, G's, C's, T's. Okay. These lines here are representing hydrogen bonds between complementary bases. Okay, so Something else that you guys should be prepared to do is tell me what the complementary bases in DNA are. And so this is hopefully a little bit of review. All right, so A is complementary to T. G is complementary to C. G, A, T. Okay. So both of these strands are DNA, right? These are complementary bases, right? Even if I didn't tell you that these were both DNA, you could tell because T is only present in DNA, not in RNA. Okay, so the cookbook is stored here, the instructions for everything. But in order to make proteins, in order to make all of the things that the inner workings of our cells, of our body, all of that happens out here in the cell. Right? And I think I forgot to mention this before, but all of this space inside the cell is called the cytoplasm. Cyto for cell, plasm, so plasma, some kind of fluid. The cytoplasm is essentially like kind of like runny jello almost, in which all of these other organelles are going to be suspended. Okay, so making the protein itself, so reading the recipe, putting the ingredients together, cooking the protein happens in the cytoplasm, but the recipe is stored here in the nucleus. In order to get the recipe out here, we first must make a copy of the particular recipe within the DNA. Okay. So that is actually the first step in this entire process. The first step is called transcription. And I'm going to flip back to the PowerPoint notes here in just a second, but in transcription, we are essentially transcribing the recipe. So taking word for word what that recipe is and copying it down. The copy itself, the little note card, is mRNA. Okay. So we'll say that this here is a gene in the DNA. Okay. This particular gene maybe is A, G, C, T, T, etc. What is first going to happen in transcription, which I'll show you an animation of in just a moment, is that an enzyme, I'll say this little block here is an enzyme, is going to read the DNA and make a copy of it. Okay. The enzyme is called RNA polymerase. Okay. ACE implies enzyme. Polymer is what it's doing. A polymer of what? It's making a polymer of RNA. So as this little RNA polymerase chugs along this strand of DNA, what it is going to do is attach the complementary bases to each other. Okay, so A in RNA is complementary to U. G, always with C, always with C. T in DNA is complementary to A in mRNA. Again, A. Right? And so eventually we are going to create this big long strand of something called mRNA, messenger RNA. And I just did a teeny little example here. Okay, I'm going to spread it out a little bit when we move along, but again, we are making a copy of one section of DNA, a gene in the DNA. The copy itself is mRNA.
I have a question.
Right. Questions about that so far?
I have a question.
Sure.
So the chromatin is going to be ripped apart before this?
Yeah, that's actually a great question. So remember that the bonds that hold these two DNA strands together are hydrogen bonds. One of the things that RNA polymerase does is it kind of like spins really fast, and it temporarily breaks these hydrogen bonds so that it can read one strand of the DNA, right? And then once the RNA polymerase passes, these two strands join back together and the hydrogen bonds reform. That's an excellent question. Thank you.
Thank you.
Sorry, one second, guys. There we go. All right, so this is where we left off on the PowerPoint here. Okay, so the process of transcription in a little bit of a prettier way than I am able to draw here on the paper. Transcription involves the RNA polymerase kind of unzipping these genes, right, breaking those hydrogen bonds just for now, and bringing the complementary bases together, RNA, right, you won't see any T's, just U's, bringing them together and joining them together with these bonds, okay? Here is another explanation of this process if you want to read into it a little bit more. And just as I did before, unfortunately, Flash Player doesn't work anymore, but I have recorded myself doing this in the past. So let's take a look at what the Flash Player would have showed us. Okay, so before I play this, what I want to point out is that this here is DNA, right? Double-stranded complementary bases, the whole 9 yards, RNA polymerase. Okay, RNA polymerase is going to bind to the promoter, right? So we don't have to know that much detail, but to a particular part of DNA. And what we can see here is it is joining together these bases to make a copy complementary to the gene in DNA. Okay, there is another sequence that's called the terminator sequence, right? Again, we don't have to know that much about the genetics, but essentially it says, oh, this is the end of the gene. RNA polymerase, your job here is done. Okay. Okay, so that is the process of transcription. Okay. Making a copy. However, this copy needs a little bit more love. It needs a little bit more processing before it's ready to go out into the cytoplasm. Okay, so the next step of the larger process of protein synthesis is to process the mRNA. Now, before we process it, we should understand that a gene in the DNA contains a couple different components. Some of these components, some of these sequences don't actually code for a protein, right? So it used to be called garbage DNA. We know a little bit more about it now. It's not actually garbage. It's kind of a tie-over of the evolutionary process, but long story short, some sequences within the DNA, within the gene itself, are non-coding. They don't make a protein at all. So in order, or before the mRNA can leave the nucleus and go out into the cytoplasm, we need to remove what are called the introns, the non-coding regions. The rest of the gene, right, so here and here, for example, consists of exons, and these exons are the actual recipe components, all right? So they are actually telling the ribosome, as we're going to see, what amino acids to bring together to make the protein. Okay, so the exons are the stuff that we want to keep in, and the introns are the stuff that we want to exit, right? I know it's exactly opposite of what you would want it to be, but unfortunately, that's how it goes. Okay, so what's going to happen in the nucleus is another cute little molecular structures called SNRPs are going to read this mRNA and essentially cut out the introns and splice together the exons. Okay, so that the final mRNA, of course, is much shorter than the original mRNA, and it only contains instructions for making a protein. Okay. Another way of looking at this, we remove the introns, splice together the exons, and continue processing the mRNA. Okay, so we splice together the exons, and two more things have to happen. First of all, we have to add a cap and a tail. Now, if you remember from the biochemistry lecture, the nucleic acids kind of have like a beginning and an end of the molecule. So they have a 5' end. So the phosphate side, the 5-carbon end of the nucleic acid. And then they have a 3' end. So this is kind of like the end of the molecule. Okay, so DNA and mRNA have a 5' end, and they have a 3' end. Okay. All enzymes, including the ribosome, right? Again, we are getting there in just a moment. Everything reads nucleic acids from the 5' end to the 3' end. Okay. So in the nucleus, we are going to add a cap to essentially the beginning of the mRNA molecule. Okay, so we are going to essentially add like a little flag that signals the ribosome that, hey, this is the beginning of the molecule. Start reading here so that you can read the instructions appropriately. On the back, on the end of the mRNA, we add what is called a poly-A tail, so a 3' poly-A tail. Poly-A implies polyadenine. We're going to add lots and lots of A's to the end. And the reason that we do this is that mRNA is constantly being broken down. Okay, so whenever you have mRNA hanging out in your cytoplasm, there are enzymes that are going to start munching up and digesting that mRNA from the end of the sequence towards the beginning of the sequence. In fact, mRNA only really lasts for a couple hours tops. We are constantly breaking it down. And so just a tiny side note here, when you get an mRNA vaccine, all right, so COVID vaccine, they're all mRNA vaccines, we are injecting mRNA, which is broken down within a few hours of receiving that shot. Right? But in those few hours, our body is doing this process. Our cells in your arm, when you get the shot, is doing the rest of this process. Okay. So again, mRNA doesn't last very long. Okay. It only lasts a few hours. And so what we do with our genes that we want to actually, make the recipe here, we want to make the proteins, we add a bunch of nonsense at the end. So we actually extend the life of our mRNA just a teeny bit so that there's actually time enough to read the mRNA and make the protein. Okay. Any questions about that? All right, so let's draw that in. Okay. Before the mRNA can leave the nucleus, step number 2 is mRNA processing. Okay. I am going to show the end result because, of course, I didn't leave myself enough room here. So I'm going to show you the end result of this processing. What does this look like? Okay. We have the 5' end, which is a cap, right? Essentially a flag that is showing the ribosomes where the beginning of the molecule is. Right? We have our sequence, right? Our mRNA sequence, which, is just a series of nucleotides here. Okay, and so on and so forth. And all the way at the end, we have a poly-A tail. So lots and lots and lots of A's. Right, and that is on the 3' end. This is the mRNA. It is the copy of the recipe that is stored within the nucleus. Okay. All of this stuff, right? These used to be exons, right? So these are the spliced together exons. So mRNA processing includes the addition of the cap and the tail, and the introns are spliced out. Okay, so all of those steps that were in the slide we just took a look at. Okay, so that is the mRNA processing. Okay, now this kind of stuff, transcription and the processing is happening all the time. Okay, simultaneously, other things that are happening all the time are happening on other portions of the DNA. Right, so that's what we're going to look at in the next slides here. But before we do, let me just point out that this DNA contains the genes for any type of RNA. Okay? In order to complete this process out here in the cytoplasm, we need to also make tRNA, right, transfer RNA, and we need to make rRNA. Okay. Now, both of the genes for both of these types of RNA are stored elsewhere in the nucleus. Okay. First of all, the rRNA. The genes for this type of RNA are stored within a more densely concentrated region of the nucleus. All right, this region of the nucleus with very densely packed DNA is called the nucleolus. Okay, so nucleolus, and we're going to see these words on the slides in just a moment. Okay. So it's just a smaller sphere within the nucleus. Okay. This is where our RNA genes are stored, and so in order to make our RNA, we do the same thing. So RNA polymerase makes a copy of, for example, this region of the DNA, which is now the rRNA. Okay. tRNA made again in exactly the same way. RNA polymerase is going to make a copy of, say, this portion of the DNA, which spontaneously folds on itself to be roughly T-shaped. Pardon me for not being a wonderful artist, but this is tRNA. Okay. So again, all of this stuff is happening all the time. Okay. We need these components in order to complete this process.
The transcription of rRNA and tRNA and the nucleolus, is there DNA in there also where they can transcribe, or do they get DNA from the nucleus?
Yeah. So that's a great question. So DNA is filling the whole nucleus. Very densely packed DNA in the nucleolus. And so this copying of the DNA in the nucleolus and elsewhere in the nucleus is making tRNA and rRNA as well. Okay, I'm trying to separate these processes because this gene is the one that we care about. We want to make, amylase or something here. But in order for the rest of this process to happen, we need to use other portions of DNA. Okay, so all of this is also DNA. Did that answer your question at all?
Yes, it did.
Okay, great, thanks. Okay. So in the nucleolus here, densely packed region of DNA, this is where we make our RNA. Okay, and tRNA is made elsewhere within the nucleus as well. Okay, once we have both of these components, they are actually able to pass through the nuclear pores as well. Okay, so the nuclear pores allow mRNA, they allow tRNA, and they allow rRNA as well. Before they do, however, rRNA actually joins together with other structures, right? So other proteins to make a ribosome, right, which is what I'm going to draw in just a moment. But before I do, ribosomes are another type of organelle. They are a very ancient type of organelle. We see them in bacteria, we see them in archaea, we see them in every organism on the planet. And it's a very ancient organelle, so ancient, in fact, that they do not have their own membrane. Right? So they are not a membrane-bound organelle. Ribosomes are made up of rRNA, which we just made in the nucleolus over here, and proteins. Okay, so proteins that function as enzymes. Collectively, when the rRNA and the proteins join together, they create this structure here. Okay, there is a large portion or subunit of a ribosome and a small portion or subunit of the ribosome as well. And essentially, these two subunits are going to sandwich the mRNA in between them and essentially read the code within that mRNA. Okay. The process of reading the mRNA and therefore making a protein is called translation. All right, so transcription happens in the nucleus, it's copying the recipe. Translation is reading the recipe and making the protein from it. Okay. These ribosomes are, of course, constructed within the nucleus. They leave the nucleus, go out into the cytoplasm, and so they're just kind of floating around out here in the cytoplasm all the time, always looking for this cap so they can start doing their job. Okay, now at a certain point, many of these ribosomes actually take the entire process here and they attach to something called the rough endoplasmic reticulum, or RER. Okay, we're going to look at that in just a moment, but the point I want to make now is that ribosomes always start off floating around in the cytoplasm, and under certain conditions, they're going to bind to another organelle, making that organelle look a little bit rough. Okay, okay. So before I draw it in, let me show you what this looks like. Again, using my super ghetto previous video here. Okay. What we're about to see is the mRNA sequence. This is the recipe. You can see that there are no T's, it's only used. So this must be mRNA. Here's the cap, here's the tail. So what is going to happen here in just a moment? Here I am explaining exactly what I just explained to you. What is going to happen here is that the small ribosomal subunit is going to be floating around the cytoplasm, and it is going to see this cap, 5' cap, and it is going to attach to the mRNA. Okay, so this shows at the beginning. The ribosomal subunit is going to kind of read this mRNA and look for something called a start codon. Okay, once it finds the start codon, we have a tRNA bring the very first amino acid into the ribosome. Okay, the start codon is always AUG, right? 3 nucleotide long sequence. Okay, and this again is signaling the very first amino acid to be brought in, transferred in by tRNA. Right, now, once that happens, or rather before that happens, the small ribosomal subunit, of course, is reading this code, and it will not begin the process of translation until it finds the AUG. So it doesn't matter how many nucleotides are here before the AUG. AUG always signals the start. Okay. Start codon binds the tRNA with the complementary anticodon, and then the large ribosomal subunit is going to sandwich all of this stuff in between. Okay. Next, the next tRNA is going to arrive. It is going to bring with it the amino acid for the next codon in line, the next sequence of 3 amino acids. The ribosome, with its enzymes and rRNA, is going to catalyze a reaction between these 2 amino acids. Okay, so methionine and lysine are going to be joined together with a peptide bond by the enzymes within the ribosome. Okay, now once this reaction has occurred, what's going to happen next is that this first tRNA, which is now empty, is going to kind of peace out. It's going to get lost. We don't need it anymore. Okay, and the entire ribosome is going to slide down the mRNA. Okay, so what we can see now is that the next codon in line is within the ribosome. The used up old tRNA is going to leave, and it's going to search for another amino acid. New tRNA is going to arrive in the ribosome, right? Again, based on this codon. And once again, the ribosome is going to catalyze a reaction between the last amino acid and this amino acid. So what we are doing is we are building up a chain of amino acids, which, as we know from our biochemistry lecture, okay, a chain of amino acids is a protein. Protein sequence or primary sequence is coded for by DNA via mRNA. Okay. Again, old empty tRNA gets lost, new one comes in, reaction is catalyzed, lengthening the chain, the entire ribosome moves down the chain again. Okay. And the process continues. Okay. Any questions about that yet? Okay, I know it's a lot. So let me spell out the steps for you, and then we're going to draw it. Okay, so essentially the words of the recipe, right? So cup versus teaspoon versus, sugar, flour, whatever. The words that actually code for the ingredients are called codons, right? 3-letter sequences on the mRNA. This here is a chart of all of the possible combinations of nucleotides, right? Therefore, all of the codons that could possibly be within the mRNA. Okay. According to these codons, the tRNA, which is kind of upside down from what we've been seeing, but this tRNA is going to bring this. So these are the amino acids that make up the protein. Okay, so here we can see the tRNA has an anticodon that is complementary to the codons in the mRNA, right? Bringing with it the correct amino acid. Okay, again, codons, part of mRNA, anticodons, part of tRNA. In order to actually translate the protein, we read the codons with this chart, and the tRNA is what is actually going around the kitchen and bringing the appropriate ingredients. Okay. The beginning of any protein, no matter what, is the start codon. Okay. AUG, no matter what, no matter whether there are 1, 2, 3, 500 nucleotides, the first AUG is where we start the protein, no matter what. Okay. And so this link right here, if you somehow miraculously got Flash Player to work, that is the link that actually shows you what I just showed you. Okay. So let's draw this in. The next step, number 3, is translation. All right, going from nucleic acid language into a protein language. So translation is making the protein or synthesizing the protein. What is going to happen is that this ribosome made out of our RNA and proteins is chilling in the cytoplasm. Okay. It sees the cap and it starts reading this mRNA. Okay, so what we do is we look at the sequence, right? We are looking for the start codon. So nope, 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 not yet. Aha, there is the start codon. AUG no matter what, right? Doesn't matter how many nucleotides are here, can be 5, can be 17, can be 500, doesn't matter. AUG is always the beginning. The small ribosomal subunit binds to the mRNA, and simultaneously a tRNA with the complementary anticodon. So this is the start codon. Complementary anticodon would be UAC. Right, and this line that I'm drawing is just the rest of the sequence of the tRNA. Right, so this is kind of what else it would look like. And opposite to this anticodon, we have the amino acid. Now, if you look at that chart, which I'll bring up in just a moment again, AUG always codes for methionine or MET. Okay, so this tRNA brings methionine because of this codon. Next, the large ribosomal subunit is going to bind, essentially sandwiching the mRNA, the tRNA, the amino acid inside. Once that is the case, the next tRNA is going to arrive. All right, we can see that the anticodon would be GGC. Again, you should be prepared to identify the complementary sequences. Okay, so this is our next tRNA. All right, so what amino acid is going to be transferred by this tRNA? Right, and we can figure that out. The way that we do that is with that fancy chart. Now, on a lecture exam or a quiz, I would give you this chart. Okay, so our codon is CCG. Okay, so CCG is pro or proline. Right? You don't have to memorize this chart at all. You don't have to memorize that pro means proline. Instead, all you have to do for me is identify that CCG codes for pro. Yep. Again, I would give you this chart. Okay, so now we have the first two amino acids, but they're still both attached to the tRNA. The ribosome is made up of enzymes in our RNA. It catalyzes a reaction between methionine and proline. Right? We know from our biochemistry lesson that this bond is a peptide bond. Okay. We know that enzymes reduce the energy of activation. We know that enzymes bring the reactants here, methionine and proline, into close proximity with each other to make it much easier for them to form a bond. Right, so that is what's happening here. Note how we just build and build and build on these concepts. Once this reaction has been catalyzed, this tRNA gets lost, right? So it's going to go back out into the cytoplasm and get another methionine so that it can help out for the next protein. Okay, the entire ribosome, small and large subunits, are going to slide down the mRNA strand. Yep. The next codon is AUC. So again, a tRNA with UAC, UAG, is going to arrive in the ribosome, bringing with it the next amino acid. I hope I didn't stop the protein here. All right, so UAC. UAC is TYR, right? So tRNA brings this next amino acid. Ribosome catalyzes the reaction, emptying this tRNA, which can then get lost. The whole thing slides down and continues with the process. Are there any questions about that? All right, so again, translation is the process of the ribosome reading the mRNA, catalyzing reactions between amino acids, which are delivered by tRNA based on the codons within the mRNA. The product of this entire process is a chain of amino acids, which we call a peptide or a polypeptide. Okay, so in purple here, this is our developing protein. All right, not done yet. Okay, which is made of amino acids. Now, there are lots of different types of proteins, as you might imagine. Some proteins are just going to hang out in the cell for the rest of forever, right? For the length of their existence. Other proteins, however, and that's kind of where we're going with this story, other proteins are going to be embedded within the plasma membrane, right? So they're going to become some of those channels, right? They could become embedded within other organelles. They could become enzymes within other organelles, or they could be released from the cell entirely. So for example, we have a lot of hormones that happen to be proteins. And so in order to release those hormones out into the blood, which would be out here, they need to leave the cell, right? So most of our proteins do not get finished right here on a free ribosome. Instead, this entire thing, mRNA, ribosome, growing protein, this whole thing is going to be moved to another part of the cell, and it is going to be embedded on another organelle. That organelle is something I mentioned before. It is actually attached to the nuclear envelope. Right? It is a series of flattened saccules that are all interconnected with each other. Okay. It is called the rough endoplasmic reticulum. Okay, and I'm going to show you this on the slides here in just a moment. All right, but again, where we're going with this is that if this protein is destined, right, its function is to become part of a membrane, part of an organelle, or leave the cell entirely, this whole thing is going to be bound to the RER. Okay, so there are little like docking proteins. We don't have to understand too much about this, but making the ribosome much, much tinier now, right? So there's the ribosome. It is still reading the mRNA, right? It still needs the recipe. It is making a protein, but now the protein isn't just going to be out here in the cytoplasm. The protein is going to be, or is going to enter into the RER. Okay, so each one of these little circles here is an individual amino acid, right? And the lines are the peptide bonds between them. Note that the ribosome continues to read the mRNA, making a protein in response. But now this protein needs a little bit more love. It needs a little bit more processing before it can go forth and, you know, complete its destiny, right? Like do its function. Okay. So the protein enters into the RER. Okay, so if the protein is within a membrane or leaves the cell, the ribosome, which used to be free in the cytoplasm, attaches to the RER. The RER is called the rough endoplasmic reticulum. Again, it is a series of flattened saccules or channels, right? So nice and folded on itself. All of them are interconnected, right? And this is actually an extension off of the nuclear membrane, right? So the RER is always wrapped around the nucleus like this. The reason it's called rough endoplasmic reticulum, right, is because it looks rough. Also, anytime you see these kind of extra words, right, rough, that implies that there's going to be something like opposite to it, right, or counter to this, right? So if there's a rough, chances are there is also going to be a smooth endoplasmic reticulum, and indeed there is. Okay, so this is the rough endoplasmic reticulum. It is covered with ribosomes that are continuing the process that began out in the cytoplasm. Okay, so what exactly happens in the RER? Well, in the RER, the proteins, right, which are made by the ribosome, are folded. Right? Remember that proteins have a primary, secondary, tertiary, and sometimes even quaternary structure. Okay, so in the RER, the first couple levels of folding or structures are going to be formed. Okay, this is the primary structure in the ribosome. This is the formation of the secondary and tertiary structure. Okay, so let me add this here. Step number 4 is happening in the rough endoplasmic reticulum. We have folding into the secondary and tertiary shape, right, or structure. Secondary and tertiary. And just like we saw earlier today, a lot of the proteins that are embedded within the plasma membrane aren't just plain old proteins. A lot of them have carbohydrates attached to them. Okay, so this gives the cell essentially like a fingerprint. Well, where are those things attached? They are attached in the rough endoplasmic reticulum. So the carbohydrates are attached if that is, you know, the destiny of this particular protein. Okay. When I first learned this, it was described to me as kind of like planting hedges, right? So planting like bushes in your front yard, just unruly bushes, just like lots and lots of carbs attached to the existing proteins. Okay, to be continued. Okay, this is kind of a prettier way of showing this. All right, so here are those interconnected RER membranes. Okay. Here is the ribosome with its mRNA, right? When signaled, right? When this particular protein has a code within it that says it's going to go out of the cell someday, the ribosome binds to the RER membrane. The protein enters into the RER, and once it's in the RER, these little green carbohydrate hedges are planted, and it is folded into its secondary alpha helix beta pleated sheet structure and its globular structure or tertiary structure on top of that. Okay. Ultimately, the ribosome is going to stop translating when it reaches what is called a stop codon. Okay. So let me go back here for just a moment. If you look closely at this chart, you'll see that there are 3 sequences, UAA, UAG, and UGA, that all don't code for an amino acid. They code for stop, right? So in this sequence, whenever the ribosome reaches one of these 3 sequences, the protein is done, right? Usually that's going to happen once it moves to the RER. Okay, so on a lecture exam, I might give you a sequence, right? You might have to translate that sequence. Okay, so the first thing you're going to do is look for AUG. Right, you're going to go back and you're going to look at this chart, right, and tell me all of the amino acids that are coded for by the sequence. And you're going to stop translating whenever you come across UAA, UAG, or UGA. Okay, if there's not a stop codon, go back and check. I'm going to give you a stop codon. Okay. If you don't see one, you may have made a mistake. Go back. Okay. So again, protein is destined to leave the cell or become part of a membrane. Entire structure attaches to the RER. The protein is finalized and the ribosome stops translating as soon as it comes across the stop codon, leaving essentially this folded up protein with carbohydrates attached. So I'm just going to draw like really branched, kind of unruly little hedges here. Okay. Secondary and tertiary shape, just a note on annotation here. 2 with a little degree sign means secondary. 3 with a little degree sign means tertiary. Right, just so you know. Okay, and carbohydrates are attached. Questions about any of that so far? I know it's a lot. All right. At this point, I want to take a moment to pause the protein synthesis process. Okay. There are a few more steps, as you might imagine. It's all very complex. But I told you that if there is a rough, there's probably going to be a smooth. And so I want to make a little bit of a tangent here. Smooth endoplasmic reticulum. This organelle is also a series of interconnected saccules that are more or less continuous with the RER, which is continuous with the nuclear envelope. Okay, so looks kind of similar, but it's not rough. The reason it's not rough is that it has nothing to do with protein synthesis. It does not have a bunch of these tiny, cute little ribosomes all over it. It is smooth looking on the outside. So it's not involved in protein synthesis, but it is involved in synthesizing and processing lipids, right? So the fatty stuff, the oily stuff. The SER, smooth endoplasmic reticulum, produces lipids like cholesterol and other types of derivatives of cholesterol. All right, remember that in the biochemistry lecture, we talked about steroid hormones, we talked about eicosanoids, all of those things can be made in the SER. Okay, so the synthesis of steroid hormones like estrogen, testosterone, cortisol, your stress hormone, right, all those things from the SER. Also, they essentially take fats and oils that you consumed in your diet and they convert it into human molecules, right? So the molecules that are used to signal or store or whatever within our body. Okay. The SER is also responsible for detoxification of different chemicals, either ones that you take on purpose or ones that find their way into your body against your will, right? So extracurricular substances, as well as things like pesticides on your food, chemicals that you're interacting with in your local environment that you're breathing in. All of these things are broken down specifically within the SER. As a result, any organs that are responsible for filtering fluids or blood within your body have a larger SER in every cell. So for example, your kidneys and your liver, one of their jobs is to filter stuff we really don't want in our body out of our body. And so a liver cell is going to have a lot more SER than a muscle cell, for example. Okay. Similarly, the liver not only breaks down toxins, which you might be most familiar with, but also it's involved in just everything, so much that's going on in your body. One of those things being energy metabolism, right? So the stored energy, either as glycogen or as fat, our SER within our liver can break those stored energy sources down and dump it into the blood as sugar if we don't have enough energy in our body already. On the other hand, if we are sitting on the couch eating candy bars and French fries, we don't need all of the energy that we are consuming. And so your liver is going to take all of that extra sugar, all of that extra fat out of your blood and store it within the cells. Okay, so the SER, particularly in the liver, is very important in those processes. Okay, now just like not every cell is equal in terms of SER, not every person has the same amount of SER either. For example, if you consume a lot of extracurricular substances like alcohol, your liver cells, kidney cells, cells in general have to work a lot harder at detoxifying your body. And so you actually have more SER if you are, for example, an alcoholic than if you never drink at all. Okay. Again, the SER has nothing to do with the process that we've been talking about all along. I throw it in there because if there's an RER, there must also be another type of ER. Okay, so that is lipids, RER is proteins. Okay, the story continues. Okay. We now have a protein in its secondary and tertiary structure with attached carbohydrates. But the fate of this protein is all the way out of the cell or mixed together with some other proteins or in some other type of organelle. So we have to do a little bit more in order to send this out into the world. The next step in this process occurs all the way on the other side of the cell. Right? So we can see this up here. RER is always wrapped around the nucleus. The next organelle responsible for processing the protein is the Golgi apparatus, and that is all the way on the other side of the cell. Right? Essentially, it is the post office, and it is ready to ship the proteins out. So in order to get the proteins from here all the way across the cell to over here, we need to package it up. Specifically, our rough endoplasmic reticulum has a membrane that is made up of a phospholipid bilayer. what we do is we take this protein and we kind of push it towards the membrane, creating a little bump, right? This little bump will ultimately form what is called a vesicle, right? So just like you can kind of play with little drops of oil on a piece of wax paper or something, and when those drops touch each other, a bigger drop forms. Same thing is happening here. Once these two portions of phospholipid bilayer touch each other, it forms a droplet of oily phospholipids. Okay, inside this droplet is, of course, our protein. Okay, so we form transport vesicles literally out of the membrane of the rough ER. Okay, then we can actually ship this little vesicle across the cell. Or as soon as it touches the Golgi apparatus, which is also made up of the same stuff, right? Phospholipid bilayer, the vesicle and the Golgi apparatus membrane are going to fuse together, dumping off the protein into the next organelle. Okay. So let's add the transport vesicle here. Okay. The protein is pushed against the side of the RER, forming a little vesicle. Okay, so here is our protein. We know it has all these little spiky carbs all over it. And ultimately, a vesicle is formed. Okay. In a few slides, we're going to talk about exactly how these get transported. It's kind of fun. Right. This is our transport vesicle. Step number 5. Transport vesicle. Vesicle is going to carry the protein from the RER to the Golgi apparatus. Okay. The Golgi is also made of phospholipid bilayer. Note, blue is phospholipid bilayer. Here and here, the RER, the nucleus, all of these things are made out of the same stuff. Right, so this is the Golgi apparatus or the Golgi body, right, depending on what source you look at. As soon as this vesicle touches the Golgi apparatus, the membranes fuse together completely automatically, and the contents of the vesicle are going to be dumped into the lumen or the opening on the inside of the Golgi. All right, so this is still our protein. It has been transported across the cell. Any questions about that? The story's almost done, I promise. Okay. The Golgi apparatus is a series of not interconnected, right, independent saccules. They're called cisterna for singular, cisternae for plural. Essentially, what the Golgi does is it's going to finish up with the production of the proteins. Okay, specifically, it is going to join together proteins that are meant to be together. Right, so in the biochemistry lesson, I told you that some proteins need a quaternary structure as well. So for example, hemoglobin needs 4 different proteins all squished together in order to be a functional molecule and actually transport oxygen throughout your body. So at this point, each of our proteins is independent, but what's going to happen is that this protein is going to be sent from this cisterna to this cisterna to this cisterna and be joined together with the other proteins that give rise to the functionality of the molecule. Okay, so number 6, for example, in the Golgi, we add the quaternary structure or shape. We trim those carbohydrates. All right, remember that the glycoproteins, so the carbohydrates attached to the proteins, form a fingerprint. So far, these hedges are just like unruly. We need to trim them to make sure they look exactly like us. They are our unique cell or our unique fingerprint. My glycoproteins don't look like your glycoproteins, which don't look like your parents' glycoproteins, right? Everyone has different shapes of these hedges on the surface of their cells. Finally, once the protein has been joined together with other proteins, the carbs have been trimmed, the Golgi apparatus is going to, once again, package up these proteins and take them to their final destination. Okay, so maybe in high school biology or something, you have learned that the Golgi is essentially like the post office or FedEx or whatever for the cell. And where that comes in is that the finalized protein is going to be dumped into a vesicle, and that vesicle is either going to be released or release the contents outside of the cell, or it's going to become a new organelle. Okay. Right, so before I draw it out here for you, what we have seen so far is the protein has gone from the RER, right, where the ribosome was finished. It was processed a bit in the RER, secondary, tertiary structure, carbs are planted. Part of the RER bubbles off, becomes a transport vesicle, which is carted across the cell to be continued, fuses with the Golgi. The Golgi passes it from saccule or cisterna to cisterna. Once the Golgi is finished with it, part of the Golgi bubbles off into a new vesicle, and this vesicle can either fuse with the outside of the cell, dumping its contents outside, or the vesicle itself can become something functional inside the cell. So let me draw that in real fast. Okay. Protein is now finished. All right, so all done. Here's our protein. Okay. Vesicles coming from the Golgi apparatus can either fuse with the outside of the cell and therefore dump their contents outside. This is a process that we will learn about on Monday, I believe, called exocytosis. So something to do with exiting cytocell. Or this vesicle can become a new organelle. Okay, so depending on what is inside here, what kinds of proteins, this can become a new organelle, or it can transport to another type of organelle. For example, this can become what is called a lysosome. Okay, if that is the case, inside this lysosome, there are a series of enzymes. Right, so if the protein that we've been working on was an enzyme, maybe it will be packaged up into a lysosome and stay in the cell to be functional. Okay, so these organelles can be lysosomes, right? So a lysosome is essentially like a mobile stomach inside the cell. Right? It is chock full of digestive enzymes, and also enzymes that keep the pH of this organelle really low, right? So super acidic. So literally it is the stomach of the cell. What can often happen, and does often happen in our cells, is that we take something from outside the cell inside our cells. So this right here. What then happens is that a lysosome is going to fuse with that endocytosed vesicle. We're getting there. And the enzymes from within it are going to mix and break down whatever that foreign substance is. So, maybe we're eating something, right? Or maybe this is a type of white blood cell that recognizes, oh my goodness, that does not look right. That looks like a spike protein from a virus, for example. We're going to take it in, break it down, and then deal with it accordingly. Okay, so lysosomes, super important. They will come up again. Another type of vesicle from the Golgi apparatus that can become its own new organelle is a peroxisome. Right? So perox sounds like hydrogen peroxide. That is where it gets its name. These peroxisomes specifically break down excess lipids in our cells. And as a byproduct, they produce hydrogen peroxide. Right? There are also other enzymes within the peroxisomes that then break down the peroxide, right? Because peroxide is lethal to our cells, right? And also to, invaders' cells, bacterial cells for the most part. So we make the byproduct and we then break it down. Okay. Any questions about this entire process? I know it's a lot. Again, so how would I test on this? On an exam, I could ask you about any of these different steps that are occurring, right? So ask a question about transcription, right? What is transcription? What is RNA polymerase? What is happening here? Can you transcribe this gene in DNA? So any of these different steps, what I recommend, and I recommend this every semester, is for you guys to draw out this process again and again from memory. Make sure that you can talk about it without your notes, that you can bring up individual terms, right, from memory, right? So when I do ask a question about codons versus anticodons, you can, in your mind, go right back to your image here and bring up the appropriate answer. Okay. Now, because we're online, the last few semesters I have been doing or kind of helping you guys to give this process a try. That is the study process of writing this out over and over again and talking about it like a story almost. Okay, and so next week I'll talk about this a little bit more, but essentially half of the class is going to essentially teach this process on a video and then give the video to me so that I can see, maybe you understand all of it, but this part right here, you're a little bit sketchy on. So let's dive into that. But also this experience is going to teach you guys how to study physiology. And I always kind of describe the physiological concepts like a story. This happens, then this happens, then this happens, and finally all the way over here, the end. Oh my goodness, we did it. Okay. I encourage you to do the same. Making flashcards about this kind of information does not allow you to see the connections and the causality between this and this and this. So to be continued, but expect that you could have to talk about this process in a video forming. Now, the other half of the class is going to have another process similar to this that we will talk about next Tuesday, I think. All right. Again, any questions about that? All right. Couple more slides here. All right. So that is the process of protein synthesis, but it is, or on the upcoming quiz and lecture exam, you aren't only going to have to talk about protein synthesis organelles, there are some other organelles that are important as well. Okay, the next one is the mitochondria. For whatever reason, it seems like this is the one that sticks with people from high school. This is the cell's powerhouse or energy center. Essentially, in order to fuel your cells, we need ATP. Where do we get the ATP from? Well, we get it from sugar. We get it from our food. Okay, so sugar, and in some cases, directly from fat and protein, but for the most part, we take sugar out of our blood and we break bonds in those glucose molecules, releasing energy, which we then use in ATP. The organelle that is responsible for getting the most bang for your buck, for getting over 30, right? So 32 to 36, depending on the situation, ATP for every single glucose molecule is the mitochondria, right? So we take glucose and we get ATP from it. As a byproduct, we make carbon dioxide. So the carbon dioxide that we are exhaling all the time, that is directly made by the mitochondria within all of our cells. We also make a bit of water, right? So we are literally making water by playing with energy, right? So cool for us. Mitochondria, just like most of the other organelles that we've talked about, with the exception of the ribosome, is covered by a membrane. Okay. However, it's not covered by only one membrane, it's covered by two. So the mitochondria and the nucleus are the only organelles that have two membranes. Both of these double membrane structures were likely other organelles or other organisms way back in evolution, and we developed this lovely symbiotic relationship, and now they are literally part of us. So endosymbiosis, if you're interested. For our purposes now, right, the nucleus must protect the DNA with two layers of membrane. The mitochondria actually uses these two membranes to create what are called concentration gradients, more on that on Monday, to make the ATP. Other kind of cool thing about the mitochondria is that it has its own plastid DNA, right? So the DNA isn't just in the nucleus. We actually had an entire other genome within our mitochondria. And historically, it has been shown that this DNA comes directly from mom, right? It doesn't mix with dad's, right? There's no mixing going on. You get your mitochondrial DNA directly from mom. So if there was ever a need to do a maternity test instead of a paternity test, you could just look at your mitochondrial DNA. In the past 2 years or so, this this understanding has been challenged a bit. So definitely something to keep an eye on in the news for sure. Okay. This is a summary of aerobic respiration. It's something that we'll talk about more when we get into muscle physiology. But just to summarize and preview, here's the blood. Here's a phospholipid bilayer of any cell in your body. Okay. Glucose is taken from the blood through a protein channel in our cells into the cytoplasm. In the cytoplasm, there are a series of proteins that essentially break the glucose in half, and we get a net 2 ATP out of a glucose molecule without any oxygen, without any fancy stuff put into it at all. But that's really inefficient, right? So we can take the leftovers of this process into our mitochondria, use our favorite, the Krebs cycle and electron transport chain, to make additional ATP. Okay. So in order to do this, we need lots of oxygen, we make CO2, we make water. Okay. More on that later. You will never have to spit back the Krebs cycle at me at all in this class. We just care about the big picture stuff in terms of this. Okay. Final group of structures for today. The cytoskeleton, right? Cyto means cell, skeleton means, well, skeleton or structural components. The cytoskeleton of the cell consists of many of these protein fibers within the cytoplasm. So what we just talked about was making proteins. Some of those proteins get made entirely in the cytoplasm by the ribosome. They don't need the RER, the Golgi, the transport vesicles, none of that stuff. That's these proteins, right? So we are constantly making new proteins in our cytoplasm as kind of a skeleton to make sure that our cells are the right shape, that they are super strong, etc. Right? Keep in mind that we don't have a cell wall like plants do. So Our cells are kind of floppy and squishy, so all of their structural integrity comes from inside, not from outside. Okay. Some of these cytoskeleton fibers can also help us to move. For us, it helps us to move cells like sperm cells can kind of swim around. Also, some of our cells have these little projections on the surface that move around. All of that is cytoskeleton. These fibers help us to separate our cells when we were making more of them. Right? So mitosis, we are essentially cloning cells. Meiosis, we're making sperm and eggs, so making 4 little haploid cells. In order to separate those cells into 2 independent entities, we need cytoskeleton fibers. Okay. Intracellular transport of organelles, which we'll go back to our diagram here in just a moment, to move stuff around in our cells, we actually have little like train tracks. Okay, there are 3 different types of cytoskeleton fibers. The first type are actin filaments. These are the thinnest of all. And essentially, when we look here, we can see these little yellow fibrous things. Those are actin filaments. We can see that they're making a little band right under our plasma membrane, which remember is the consistency of vegetable oil, right? So we need a little bit of a shell on the inside of our cell to push that membrane out so it doesn't just collapse in on itself. This provides mechanical strength to the cell. It helps to attach the proteins that are spanning the entire phospholipid bilayer. And in cell division, so meiosis and mitosis, this is going to help to separate those cells. Also, something that you are going to become very familiar with in just a few short weeks, actin filaments, even though they are the thinnest, are responsible in part for moving our entire body, our entire organism itself. What we're going to see is that we have myosin filaments, which attach to our actin. They have these little heads that rotate and they essentially grab onto the actin and they pull it. They grab onto the actin and they pull it. And so when millions of these things are pulling on actin at the same time, our muscles are going to shorten. And so we actually pull our limbs, right? Pull our limbs at our joints to move our entire body. So again, more on that later. Intermediate filaments. These are intermediate, they're in the middle in size, and they again are going to provide support and structure. Specific examples here, keratins. Right, so you might have heard of keratin because there's keratin in your hair and your nails. Also, we have keratin reinforcing our skin. Okay. Lamins, these further reinforce our nuclear envelope. So we can see in this fluorescently dyed pictomicrograph, this blue structure here is the nucleus, and what is highlighted there, what is stained with this fluorescent dye, are the lamins, right? So we keep that nuclear envelope spherical, collapsing on itself. Okay. And finally, neurofilaments. We have individual nerve cells or neurons that extend all the way from our spine down to the tips of our toes, right? So like 3 and change feet long individual cells. Now, these super skinny cells have to pass through a lot of different tissues and a lot of different, manipulations. And so it's really important to reinforce the axons, right? The portions of these nerve cells so that they don't kink, so that they don't pull apart in that really long length. Finally, we have microtubules. The name kind of implies that they are the smallest, but in fact, they're the largest of all of these cytoskeleton fibers. I don't make this stuff up, I'm sorry. But what we can see here is that each of these is a little chain of protein, and they kind of swirl together, leaving a hollow core. So they're actually, a relatively large diameter, all things considered. Let's see. Not going to worry about plant cells, but essentially our cells are constantly making new microtubules, right, with our ribosomes, right? And these microtubules are organized with something called a centrosome, right, which we're going to look at here. in the lab, I believe, but the centrosome essentially is making these new tubules and then breaking them down. We don't need those ones anymore. We're going to make new ones, and then we're going to break them down. We don't need those anymore. All right, so what exactly do these do? Mostly microtubules are involved in movement, right? So first of all, they can help organelles move within a cell. And so essentially they create little train tracks, allowing little molecular structures called molecular motors, like this guy right here, to literally latch onto an organelle and cart it across the cell. So I'll show you this in just a moment. This is one of my favorite little guys in biology, but essentially these molecular motors use an ATP to move their cute little legs. All right, so one ATP moves a leg, another ATP moves the next leg, and so on and so forth. And so they can actually walk down the microtubule. All right, so dynein is this little molecular motor. Okay, why exactly is the molecular motor moving? Well, we can see here that it's latched onto a transport vesicle. All right, so when I said that vesicles have to go from the RER all the way across the cell to the Golgi apparatus, what's actually doing that is a molecular motor walking along microtubules. And I really want to show you this because it is just fantastic. All right, so what we're going to see, these are microtubules being created, brand new all the time. All right, so building the microtubules in the cytoplasm by free ribosomes. And now we don't need it anymore. We're going to break it down. And this little guy is a molecular motor, adorable little Atlas guy carrying this big old vesicle on his shoulders. Every little movement of his foot is due to ATP. Right, so hopefully you guys appreciate that as well. They're just so darn cute. All right. Also, microtubules can move an entire cell. Okay. We can join together and bundle together a lot of what are called a lot of these microtubules covered in a membrane and call it a flagellum, right? Or flagella for plural. So this is a flagellum. Okay. Note that the flagellum is attached to the plasma membrane with this unbelievably complex little grouping of proteins. Right? There is a little hook or a little kink within these microfilaments. And essentially what is happening here is that these mechanisms, these protein mechanisms, are going to spin this rod part of the flagellum. And because there is a hook, essentially, like as this part is spinning, it's going to flip around in a snake-like undulating movement, right? And when that is happening, it is essentially propelling the cell through fluid. Okay, so sperm tail is a flagellum. Okay. Finally, these little hair-like projections are called cilia. They're essentially really simple flagella, and they can often cover the entire surface of cells. What we can see here is a type of epithelial tissue, which you're going to learn about so very soon. Stay tuned. On the lumen side of these cells, right? So the lumen is just like an opening in any structure, any organ. So lining your entire trachea, your entire bronchi, we have these epithelial cells, which have lots of cilia. We also have these little goblet cells, which are secreting mucus all the time. So when you breathe in all of the pollen-laden air from the outside world into your lungs, those little pollen grains will get stuck in the mucus. Okay, so they get stuck in the mucus. We don't want all that pollen going way down into our lungs. And so what the cilia do is they create these little wave-like motions and they push the pollen stuck in mucus up out of your lungs, up and up and up, so that ultimately you can swallow it and rid your body of those pesky pollen grains. All right. Similarly, cigarette smoking and everything, all of those particulates are being trapped and brought up and out of your lungs. Now, of course, if you do smoke for a long time, all of that tar and extra gunk in the cigarette smoke gums up your cilia, so they can't actually get the stuff out of your lungs anymore. So you actually have to cough to rid your lungs of particulates, right? Thus, smoker's cough. Okay. And that is the end of our new content for today.